How's the microphone in the back? It always sounds very loud up here. Okay, good. Okay, let me get started. Thank you all for coming back for these Saturday morning lectures by Luca and me. It's myself, it's the last lecture of this school. And as promised, I will try to make the bridge from theory to therapy. So I did from ecology to evolution over the first two lectures, from lysis to latency in the third lecture, and today I will aim from theory to therapy. And I want to acknowledge at the very outset that this is joint work. The theory side is done by Dr. Joey Leung, a PhD in physics, who's a postdoc in my group, with some additional input from Devika Singh, and I'll show some late-breaking results from a new PhD student, Rogelio Rodriguez, later in the talk. And all the experimental work was done at the Institut Pasteur, and primarily in the hands of Dwayne Roach, who was a former fellow at the Pasteur Institute, is now assistant professor of biology at San Diego State, and the lead is Professor Laurent Debarbieu with input from James DeSanto. Okay, so to get started, I want to provide a little bit of context. Right? Why is it that I'm going to give a talk on theory to therapy when I'm talking about viruses? Don't we usually want to get rid of them? Why am I thinking about using them perhaps in some form of therapeutic approach? And the reason is described in some sense by these three slides above. These banners list what are termed from the CDC, the Center for Disease Control and Prevention, the biggest drug-resistant threats. And you'll probably recognize some of these things here. All these bacterial pathogens can infect humans, causing all sorts of issues. And the worrisome part is that given our use of antibiotics to treat these diseases, and also the use of antibiotics in all sorts of settings, from industrial settings and farms and so on, there is now the proliferation, evolution and proliferation of drug-resistant variants of these pathogens. In other words, you add an antibiotic, and yet that pathogen is resistant to it. It will not die through the application of that drug, and sometimes multiple drugs. So those are called multi-drug resistant pathogens. And once you start to have a multi-drug resistant pathogen, then the options don't start to look so great, right? Because you can't treat it with your conventional antibiotics, then a simple cut or wound can become all the way to something that could be fatal, or the available antibiotics themselves have secondary effects, cytotoxic effects, so they're not going to be good for you to take in the first place. Maybe you'll get better from disease, but there's going to be a lot of secondary consequences. So there's quite a number of these, and there was an O'Neill report put out in the UK which tried to assess the potential scope of this problem. And here on the left is the number of deaths attributable, according to them, estimated due to antimicrobial resistance every year. And they claim globally this is about 700,000. And they make the claim that by 2050, if left unchecked, that this will exceed deaths by cancer. This has been somewhat controversial, but I think the major point to take away is that right now it's a threat, but it could become a genuine crisis moving forward. And he has another point to make, and this committee had another point to make, which is that at least the investment, at least in the United States in terms of NIH, relatively speaking, given the scale of this threat, has been disproportionately to other diseases that don't have this large-scale potential mortality and morbidity influence. And part of the reason why there's this gap is that until just recently, there have been no major new types of antibiotics developed over 30 years. Sure, there have been variants, but there just had been a major gap in the antibiotic pipeline. So we have this issue where there are pathogens, they've evolved resistance to the use of conventional antibiotics, often multi-drug resistance. And so what are we going to do as a way to begin to treat or consider alternatives to conventional antibiotics in treating these infections? And one of the things that people have turned to are viruses, specifically the viruses that I've been talking about in the course of these lectures, which are phage, viruses that infect and lyse bacteria. And this is not just a theoretical issue, but one that has become increasingly an applied issue. Europe, and I'll talk about some examples in a moment, but in the United States, although these are not FDA approved, they are permitted in compassionate use scenarios in which no other treatment options are available. And these are two examples, one from this gentleman on the right, who was infected by a multi-drug Acinetobacter baumannii infection, was in a coma, and reached out both to Army research labs and academic labs, and they identified a phage that was specific to the strain in combination with antibiotics, ended up curing this individual, and now together he and his wife, both professors, have started a new phage therapeutic center at UCSD called IPATH. This is another example, Paul Turner, professor of ecology and evolutionary biology at Yale. Paul, you know this Paul very well. This individual was a dentist in New Haven who had had an infection that had led to after a surgery that led to a fistula in his lungs, which had become infected with Pseudomonas, a drug-resistant Pseudomonas, and it was untreatable at the time. They ended up identifying, for reasons I'll explain later, a phage that infected in combination using phage and antibiotics in a strategic way. We're able to cure this infection, and this has become the basis of quite a lot of new work, and they've now done this in multiple patients successfully, and I'll talk more about that too. So the individual examples, I think, provide some positive hope. I mean, these are use cases where you're taking a phage, which is, it can infect and lyse a bacteria, and using a virus that infects a bacteria to help a human recover from a very serious, if not nearly fatal disease, or would have been fatal disease. And part of this is because microbes get sick too, but if the microbes are bad for you, if you can make somehow that microbe get sick or eliminate the microbe, that could be beneficial. So in an effort to try to scale this up, these individual cases are good, but you need systematic evidence that this works in controlled trials where you have a branch in which you don't apply phage and a branch in which you do. And so this was announced in September 2015, the first multi-center clinical study of phage therapy in burn victims. And you can see here they have what is standard care. Burn wounds are often infected, sometimes by polymicrobial communities. So they had an arm in which if the burn was infected by E. coli or Pseudomonas aeruginosa, they had phage specific to each type, and they would compare it against the control and see if there was improvement. And a year later, and I heard some of this at conferences, there had been an issue because they were trying to bring about 200 plus patients in, and instead what happened, they only recruited 15 of the 220 patients. With 15, you don't have the statistical power to really say anything, whether or not, even if some work, you don't have the benefit. And part of the reason there, this was called this beleaguered phage therapy trial presses on. Some of the barriers is that these individuals were developing phage specific to one microbe, but because the control was meant to be only with respect to phage of E. coli or phage of Pseudomonas, as soon as the burn victims had polymicrobial infections, they had to exclude them from the trial. So they ended up having very few examples of the scenarios in which they thought this would work. The other reason that I think there's been skepticism about phage therapy, there's many reasons to be skeptical, is that at the very origins of this field, of the discovery of phage, Felix Darel already essentially thought that what one should do with phage is use them clinically. And at the very outset had done these trials to essentially add and give phage to victims with or patients with bacillary dysentery, and claim that this helped cure them of dysentery back in 1926. Right? So if it worked in 1926, why isn't everyone using it? And so why are we revisiting it? And there are many backgrounds to that story. Part of it is the rise of antibiotics. Antibiotics were very effective. Also, phage understanding in the 1920s, 30s, 40s, et cetera, was not where we are now. So sometimes these purified phage stocks really contain lysates, maybe even toxins. Sometimes they mix with antibiotics. There's all sorts of other stuff in there. So it became, let's say, quite noisy and hard to get a handle on whether or not phage really could be used in a therapeutic sense to eliminate bacteria in a patient. So at the end of this article, which I think is provocative and should be read and taken seriously, one of the commentators said, well, it's not like there's been some transformative development or technology that it means it's open season on phage therapy. And in other words, what this person is saying is, yes, the need is greater. We have more multi-drug resistant infections. There are more of these antimicrobial infections going on. So clearly we need some alternatives. But what's different? If it didn't work before, just because the need is greater, what's going to make it work now? And I'll mention one which I'll return to later. One of this is, I think, really something transformative in the way that people think about phage therapy by Paul Turner and his group. And as I said, I'll describe it at length later, but it's a synergy between phage and antibiotics. And the idea is that many of the reasons why bacteria can be multi-drug resistant is they have these efflux pumps. So you put in antibiotics, and the bacteria just pump it out. Okay? However, in some cases, and this is not just in one particular case, but certainly in Pseudomonas, but there are other examples in which the efflux pump is also a receptor for phage. So phage bind to the efflux pump. So if one adds both phage and antibiotics at the same time, those bacteria that have the efflux pump will be infected and killed by phage. And if there's a mutation that removes or blocks the efflux pump, now they won't be killed by phage, but if you apply antibiotics, they'll be killed by antibiotics. And so Paul systematically thought through this and realized this could become an evolutionary trap, which if you add both, you're going to have a much higher likelihood of success because the one-step mutations are not going to increase the ability of these bacterial mutants or the wild type to escape from the therapy. And so that's what they've done, and they've now done this multiple times and keep elaborating on this idea that there's really a synergy. If you use two rather than just one selective force, you have a much better outcome that's more robust. And I'll put another potential synergy, I'll put it in parentheses, because I think there's quite a lot more that we have to do, and that's one having to do with a different kind of synergy between phage and the immune system. And that's what I'm going to try to talk about today, in trying to go from theory to therapy. So I'm going to try to get there in three parts. First of all, I'm going to remind you, but more briefly, because I already gave those lectures, about how virus ecology and evolution can inform what we may be doing clinically if we want to think about a phage therapy that's both effective as well as robust. And some of the dangers and the limits to the use of phage therapy, given what we know about viral ecology and evolution. And then I'm going to try to introduce some new principles underlying what I think is an immunophage synergy when we move from these in silico, in vitro settings into a setting where there's a within host. So there's an environment that is the host, and that's going to change some of the dynamics. And then I'm going to try to put these into action and show the relationship between some of what these models are doing and data from a murine model, mice model, in which the experimentalists have modulated some of the features in ways that begin to probe the limits to how effective phage therapy can be given different host contexts. So again, ecology, evolution, some more theoretical principles, and then going all the way to the application side. Okay, so this part I can do relatively quickly because you've already heard some of these lectures. I'm going to talk about the limits of virus microbe ecology in dynamic elimination of hosts. In my first lecture, I introduced multiple versions of these models where there's some resources, there are hosts, there are viruses. Critically, the viruses are obligate parasites. Although we may want to use them therapeutically to get rid of their host, that is not necessarily what they want to do or what has been happening in terms of their interactions, right? And so I made the point that when viruses and microbes interact, it's not as if the predators kill the prey. Just because they're killing individual cells doesn't mean they're killing entire populations. In fact, they tend to coexist. I also showed you that if we incorporate many of these microscopic mechanisms, you recall that that feature of coexistence, maybe oscillatory coexistence, was robust to many microscopic models. Right? I then showed you that even though in the lab setting that these viruses and microbes could coexist, and maybe, yes, the virus could draw down transiently microbial abundances, but that if one waits long enough, you see the emergence of resistant types, and then, yes, you added the virus, the host, it was reduced, but then a mutant arose, and now the host density is back more or less to where it was at the beginning, and the virus and the host are oscillating. You haven't eliminated, through the action of infection and lysis, the target microbial population. So that, in essence, gives you a summary of what I did in the first two lectures. But now I'm going to go in a different direction and say why that is relevant if we're going to think therapeutically. And the reason it's relevant is because that eco-evolutionary theory and experiments provides, I think, some caution if one wants to go and try to use phage therapeutically. And this is, in some sense, why I've been very hesitant to get into this space, because it's hard to imagine how you're going to take an obligate parasite and get it to eliminate its host robustly. Yes, viruses can kill individual cells, but they coexist with host populations. And because of evolutionary dynamics, you can get the evolution of resistance, restoring more or less the same original microbial density. So how does one deal with it? How does the field deal with this issue? The central methodological approach has been to resort not just to one virus type, but to cocktails, okay? And here, in this context, what I mean by cocktails is a combination of different viral types, right? So instead of putting one virus in the mix, you're going to put multiple viruses in the mix. And there are many ways to generate these. First of all, the idea could be that if you have a cocktail bacteriophage, each color denotes a different type. Then it could be that for some cells, they can't even get in. But for this cell, it can. Whereas other cells, the virus can get in, but now somehow intracellularly is blocked. So different phage may be able to either get in, target, and effectively lyse different subsets of the population. So if you have enough coverage, you have enough different types, maybe then that can eliminate the microbes. I'll point out that these things are available if you go to Georgia, not the Georgia in the southeast United States, but Georgia from the former Soviet Republic. This is OTC phage, over-the-counter phage that you can buy. The folks have looked at some of what's in these OTC phage, and the results have been a bit mixed in terms of what it says on the packaging and what actually is contained in the material. There's an analysis here by Sean and McCallan. And certainly there's been an investment also of trying to use various forms of reengineering techniques to mix and match, let's say, tail fibers or receptor components so that the viruses can target different sites and then maybe use effective viral cargo to try to target a more diverse set. So certainly, there's been a lot of effort done here. Yes?
So, is this something which is done for the human immune system, not to attack this virus?
I'll get back to that in the sense that I'll show you data at the end, and it really, I think, depends on the site of entry. But one could worry that there's an immune response which is going to eliminate the effectiveness once you actually go to that setting. Can we wait just on that question until I get there? But that's something that we worried about, and I'll explain why, at least for certain phage, we think it's less of a problem than has been thought in the past. Yeah. But if it happens too effectively, then that's another reason why it wouldn't work. That's right. Okay, so here we have cocktails. We have, instead of thinking about one virus as a therapeutic agent, we're going to think about many. But I would argue that if one thinks about the dynamics of virus-microbe communities, which is what I got to in the second lecture, that also provides a counterpoint to effective phage therapy. So yes, viruses can kill individual cells. So if you cover more, it can cover more, but not necessarily all. And there may be trade-offs. You're going to have to decide which of your particular viruses to use, and you'll have fewer targeting any one particular bacterial target strain. And if you get the wrong viruses, which I will talk to at the end, it won't work at all. Right? So if you haven't necessarily targeted to what's present, that also won't work. Even if you have, I mean, I showed you before that there can be reasons and trade-offs that you'll have now a complex community, but it'll still coexist. It doesn't necessarily mean just because you have viruses that can target all the variation there that all the microbes will die. You may just get a different, more complicated form of these Lotka-Volterra-like dynamics playing out at the community scale. And likewise, I think you just raised the point, it's a more complicated Luria-Delbruck experiment in some sense, because here you have microbes. If they evolve resistance, it has to be to against multiple, not just one type. But once that happens, then you're going to have lost all the effort and the benefit of this research of this particular cocktail, because now you have a new type that you haven't accounted for. Right? So there's various reasons why I think even when we go to cocktails, there's reason to have some caution. And this, again, is I think I've just summarized in a short period of time and gone a little bit quickly, but part because you've heard this before, so it's easier to follow the arc, why I was quite hesitant about going and thinking about therapeutic use of phage. So I'm going to now tilt and tell you why I changed my mind, and part of that is to begin to think about the context within which one is adding phage. So this will be on theoretical principles underlying immunophage synergy. And to begin, I want to show you some empirical data. This is work by Laurent Debarbieu, our collaborator. This is 2010, well before we started interacting. And he had been interested in trying to add viruses of Pseudomonas in this acute pneumonia model, where one gives a otherwise lethal dose of Pseudomonas aeruginosa, which in a couple days will kill mice, right, through an acute pneumonia. I mean, they sacrifice the mice before they die of the disease, but they obviously begin to show distress, and so then you have to sacrifice the mice. This is the control arm and the phage arm. And critically, they've been using a bioluminescent strain so they can track the time course of infection in the lungs of the individual mice. So what you're seeing here as 2 hours after application, 4 and 6 hours, and you can see with the red colors being higher intensity, an increasing load of this pathogen. In other words, there's an infection, it's quite active. Different replicate animals, the phage treatment, yes, there's still some, but you can see there's a contrast here in bioluminescence between the control and the phage-treated arm. This is after 6 hours. These are survival curves, percent survival of cohorts, where here is the control with no phage applied. Here is when phage were applied in a 1 to 10 ratio, and here is when they were applied at 1 to 1 or 10 to 1 ratios. So the point here is that by adding phage a few hours after the initial exposure to Pseudomonas aeruginosa at sufficient densities, you go from 100% mortality to 100% survival. And I think that raises a lot of questions. Again, these are viruses that are obligate parasites of this Pseudomonas aeruginosa. How are they eliminating, given everything I just told you in the first part? And this is a single phage, it's not even a cocktail. How is it possible that this could happen? How is it that the phage is able to somehow help or directly eliminate this target microbial population? And I think one of the most important insights in steps forward here conceptually was a paper back in 2004 by Bruce Levin and Jim Bull. And in this paper, they proposed a mechanism by which viruses and microbes would interact, but in an ecosystem context. So in other work that I do, we think about marine ecosystems in which we have to think about dissolved organic matter and zooplankton and other features. So those don't apply, but different components apply, specifically the immune response. So what they envisioned, and if you look at the top part here, this is just the standard phage-bacteria interaction that I've talked about, where phage inhibit the growth of bacteria because they're killing them, leading to more phage decay, bacteria can reproduce. And you get these sorts of models, which I've used the tau to denote the fact that these are delayed differential equation models. So here we have cell division. I haven't shown you the resource layer, but it's resource-dependent cell growth. Infection. The infection removes virus particles from the environment, and tau times later, there is some sort of lysis, and here I really, though they didn't include it there, I don't think I should have included a little bit of the discounting for the fact that some of these cells might die. Viruses decay. They then added this new component, an immune response, with two features. One is that the presence of bacteria stimulates the immune response. So here you see there's a growth rate of the immune response, and it is going to increase as there are more bacteria present. And also there's an inhibitory path that immune response and immune cells can eliminate or kill bacteria. Right? So we've taken a standard virus ecology model and put in the ecosystem in which we have this immune response, which will be stimulated and kill the thing that is stimulating it. Okay. When you take this model, I want to walk through, though it's a little hard to see. So let me just stand here so I can take a look at it. These are two different scenarios, without phage and with phage. The point that they made is that if you notice here, this is susceptible, resistant, which I haven't broken down, and immune response. Susceptible, resistant, immune response, phage. So they've imagined that there are actually going to be two kinds of microbes, both susceptible and resistant. And in the absence of phage, what they find is that the susceptible cells go above what they call a threshold, and at that moment, they assume that the infection is so severe the animal or organism would die or succumb. Whereas their claim is that if you add phage, it eliminates susceptible cells, and yes, selects for resistant cells, but the immune component is then able to control this resistant population. So they have a contrast. We took a look at this, and it turns out we worked not knowing all these details, and then realized that what we're doing was really extending the work of Bull and Levin. But I want to explain something, and the reason why we thought the extension was necessary, because there's something unusual. If I move this threshold up just a little bit, instead of putting it right there, if I just put it up a little bit more or change the parameter, you see that you don't need the phage to eliminate the bacteria. The immune system does it fine. And in fact, if you don't put this arbitrary threshold, in this model, the immune system keeps ramping up and up and up until it's so high in density that it's going to inevitably clear the infection. And in fact, if you look at the time here, adding phage seems to extend the infection to even a longer time. So we were a little bit puzzled by this and felt that there were some things that could be worth changing, that this didn't capture all the likely feedbacks in the system. And I want to tell you the two feedbacks that we then added. So we've, in effect, by a process of first, in essence, creating a model, not realizing that Levin and Bull had done this, going back and comparing, we realized that what we had really done was taken a Levin and Bull model in which we have bacterial growth, lysis, viral release, decay of virus particles, immune killing. So this is more or less the same thing, but I'm getting rid of the delay differential equation part. And we added two features. One, which is that the immune response doesn't keep going up and up and up and up without bounds. So we imagined that there was a carrying capacity in the level to which the immune response could grow. And the other was that we added a negative feedback loop here, which is that bacteria can initiate density-dependent responses and defenses, for example, biofilms, virulence factors, and so on, to evade the immune response. And this is part of the reason why one has to have antibiotics or phage in the first place. If people get infections, there is an elevated level that the immune response on its own can't always deal with. So these are these two negative feedback loops that we added. So let me examine what happens in the model with these two negative feedback loops. So first of all, we can probe it by eliminating components. So it's a tripartite system. We have phage, bacteria, and the immune response. So in the model, I can eliminate the immune response, and now I'm back to regular phage-bacteria ecology, in which in the absence of these evolutionary mutations that give rise to resistance, we expect coexistence, potentially with large oscillations, but we'll expect coexistence. So when just phage and bacteria are there, phage don't eliminate the bacteria. Okay? So again, I'm running the model without the immune response here. Okay? I can also run the model without phage. And now, unlike the case where the bacterial population ends up being eliminated, what we see is that the bacterial population increases, stimulating the immune response, but it saturates, and now this bacterial population is high enough that there are these density-dependent defenses, and they, even though there's an activated immune system, it doesn't eliminate the infection. In other words, there's an active infection. Yes, the immune system is alerted, but it doesn't lead to clearance. So what's nice here is that we have a component in which there are infections without treatment, and also if you don't have an immune system, the treatment itself on its own isn't enough. But when you combine the pieces together, we find that the bacterial population, because of the deficient of phage, is oscillating, so when it's in these peak moments, it's stimulating the immune response. But because of the phage transiently reducing densities, in one of these troughs, one of these low periods in bacterial abundance, we have an activated immune system that then can eliminate the target bacterial population. So in other words, the elimination of the bacteria is due to the immune response. In fact, if you notice here, the phage disappear before the bacteria. So this is what we term an immunophage synergy, which arises as a result of density-dependent feedbacks between these two components, phage and the bacteria, interacting with the immune system. So the immune system, right, so there's going to be an inevitable carrying capacity, and I'll talk more about which components of the immune system we're really considering here. And if you notice all these time scales, these are a little bit longer, but in the experiments and in these models, we're talking usually about a few days, not weeks. So as you'll see, we're moving in the direction of the innate immune response and not the adaptive immune response. Yes?
And you increase the immune response, you increase the capacity of the immune response, for instance, with fragments of virus, something so like a vaccine?
Yeah, so there are different ways to think about these immune response components. So certainly there are efforts to do immunotherapies, which are to either boost immune systems or modify immune systems. If it had been exposed to Pseudomonas in the past, then it would have already had a memory of that, and we would have had an adaptive response. Here, we're assuming a naive individual which hasn't been exposed before. So in particular, we're thinking about the innate response for a number of reasons, but I'm not assuming that it's already been exposed, because in that case, I should point out that in this model, if the immune response had already been activated, it could already eliminate the bacteria even without the phage. Right? There are lots of exposures we have, either because the dose was too small or the immune system was already elevated. And so there's, at least in this model regime, in which if the immune system is relatively naive and there's a large enough infectious dose, then it can't clear. But for other conditions, it can clear. Yep?
On the bacterial shyness, yeah, that's both the resistant and the...
I'm going to get into it. So at the moment, I just have susceptible.
Okay.
But I will tell you what happens when you add the resistance, because I think that's one of the major issues that we want to struggle with and try to resolve. Okay. Right, so now I just want to point out again, why does any of this matter on life history traits? I spent some time saying that the life history traits matter. So does this work for every particular phage? And the answer is no. And without going into all the details, we analyze this model and thinking about, do phage decay quickly or slowly? Do they absorb at some high rate or some low rate? And what you're seeing here in these heat maps is the predicted bacterial density and viral density. And we predict there should be different regimes, 1, 2, 3. In regime 1, we think there should be extinction. So in other words, we have to have long resident phage that absorb very effectively. And we predict down here if we have fast decaying phage, and that could be due to the immune response targeting the phage, or just because they get eliminated very quickly in whatever their the target organ is or part of the body that you've added them. So if they don't absorb quickly and they decay quick and they decay fast, then we expect bacteria to persist and the phage to be eliminated. And we also predict this very large regime in which we think there should be coexistence. But when we run the model, you'll see that there's quite a lot more elimination than we expected. So we made a prediction from the model, we ran the simulations, and it turns out it seems to do better than we thought. Right? So this is based on these coexistence equilibria. So there's a discrepancy. And the reason is in this regime, what happens is that, yes, there's a coexistence that we predict, but it's unstable. And the instability leads to larger and larger oscillations that over time we think the immune system resolves. So by looking at the stability of the fixed point, we revise our predictions and we end up getting in accord with the theory, which is basically that for a large range of phage decay rates and phage adsorption rates, we expect elimination of the bacteria given this tripartite interaction.
Does that mean that I can trust that in the blue area there is discontinuity in the density?
Yes. Yep.
So it's a sharp.
Yep, the sharp transition. That's right. Yep. Yeah, and also, yes. So, yes. And also because the phage have to depend on the bacteria. So wherever there's no bacteria, there's also no phage. Right? So we have a sharp transition here. Right. So you can see that this regime is bad because it doesn't work at all. This regime is good for different reasons. And here we have an intermediate case in which we get this coexistence, which is not desirable from a therapeutic perspective. Okay, so that's the concept. And just to put it in the schematic, what we're imagining is that there's an exposure to a pathogen which increases the density of the pathogen, stimulating the immune response. But unlike in prior work, we're envisioning that at a certain density, this actually inhibits the ability of the immune response to clear the infection. If one adds, for whatever reason, whether through biofilms or through virulence factors, if one adds phage that then reduces density of the bacterial host, which allows the activated immune response to target the bacteria, leading to the elimination of both the bacteria and ultimately the phage too. Right? Okay. And this is a schematic of what I just described. And we thought at this point this was a nice result, and we tried to send it out, and we sent it to one journal which said, well, this all sounds very good, but you haven't shown that this in vitro model works in vivo. And again, as I've been here for a few days, I do not have a laboratory. I used to have a pipette, but I gave it away. So this is a little bit hard to ask of us as a theory group to then do the in vivo version of these experiments. On the other hand, I was interested to see if this would work. Also, if you're going to start to do models of therapy, then one would like to see that this actually can go in that direction. I mean, there's an applied question here. It has some fundamental issues, but there's certainly an applied question. So let me tell you now what we've done in this direction. And this will be where we go from models to mice, thinking about ways in which one can use phage to have curative treatment in immunomodulated mice. So we're going to now see the results of what happens when the mice have some sort of variation in their ecosystem, in other words, in their immune context. And the beginning of this, since you had a beetle slide, there's the little beetles there, I get by with a little help from my friends, and I went to the Virus of Microbes meeting in 2016. It's a biannual meeting every 2 years, and you'll probably recognize the Liverpool scene. You might not be used to seeing the giant phage on top of the cathedral, but they put it there for the purposes of this illustration. So I went to this meeting in particular because they had a phage therapy focus day. And I figured that it would be a good idea for me to go have a poster on some of the theory and potentially meet some folks who are interested in the similar approaches. And it turned out there were. One of the first speakers of the day was Dwayne Roach, and he was studying the effect of phage therapy, or the effectiveness of phage therapy, using a phage PAC-P1, a phage that Laurent de Barbeau had been working on for quite some time, which had been shown, as I showed you, to prevent fatal acute pneumonia in vivo, against a multi-drug resistant Pseudomonas aeruginosa, which would otherwise cause fatal acute pneumonia in mice. But their focus was a little bit different. That had been shown before, right? Their focus was on modulating the immune status of the mice. And you can see why that would be of interest to me. Here's an example of saline mock treatment, phage therapy. Here you see the active infection. Here's curative treatment. Saline, 100% mortality. Phage, 100% survival. These are the bioluminescent curves. This is the phage treatment, which approaches and maintains at levels of detection within 1 day and stays there, and there's no recurrence of the infection later. And these are the saline mock treatments until the point that the animals succumbed. Okay. And again, this was known. This was back in 2010, and other variants of this story had been shown that, yes, there were conditions where you could use phage and it would eliminate bacterial infection in vivo. This is the moment where I thought our bit of a crazy idea could actually work. Here is saline treatment, 100% mortality. And now this is a different mouse background, this anti-GR1 treatment, which I'll explain later. Saline and phage. So here we have a mouse, which we're going to compare saline treatment, no therapy, to phage. And you'll notice all the curves do the same thing. Phage has been added, but 100% of the mice die. These are the 3 curves in which there's more or less no change in bioluminescence, with radiance, in terms of measuring the increase in pathogens. Okay? So this suggests that they've added phage, same phage as before, something has changed about the mouse, and now phage therapy doesn't work. Okay, so I'll try to explain what the difference is and how we might think about it. So we began to talk, and they said, we suggest that we could build a model to actually try to probe this and explain some of their kinetic curves and some of the outcomes. And again, as a theoretician, you think, well, we've built this in vitro model. It's certainly going to work in vivo. It turns out that's not the case. So one of our first challenges was theoretical, to really build and bridge the gap between these in vitro models and the in vivo context. It turned out that editor who rejected our paper the first time was probably right, that there was a big gap there, which is why the first version of our paper turned in the Journal of Theoretical Biology. But the other one is in, as I'll explain, is cell host microbe. It's a different context. What did we do? I took the model that I just described, bacteria, phage, immune. We collaboratively identified the relevant in vivo parameters and then turned the dial, ran the model. Hours post-infection, this is the density in terms of phage, host immunity, sensitive bacteria. And at 2 hours, we add phage. And you notice our model predicts a miracle cure. It just goes off a cliff. Right? Given the densities of phage they were adding and the absorption rates and burst size and so on, we predicted almost immediate efficacy of this model. And not only that, it didn't really even matter about the immune response. That the doses they were using, if this were a well-mixed in vitro system, would have been so high that it seemingly we would have eliminated all the bacteria just at the start. Now, That doesn't seem right. So that seems to be a challenge. What's the gap? So we have to explain that. The other thing that it's going to have to... Well, at this point, it will decay, but there's only 3 hours. It turns out the decay time was about 12 hours, so they have this big boost, and yes, they would go away, but the bacteria is long since gone. So that's a problem, right? Yes, eventually it would go away, but it's already done its job, which is not the case, right? This is not at all what the kinetic curves look like. And the other question is, what are the effector cells, right? So we can actually begin to diagnose if there's a synergy, not with just general immune response, what are the particular components that are responsible for the synergy? So I'll try to explain both of these next. So to go back to what the model described, as I've shown earlier in the week, when you have in vitro well-mixed systems, things are being stirred in chemostats, then it may be a good idea to assume that as phage density increases, that the functional response or the effectiveness of lysis is going to go up proportionately with phage, because they're in contact with all the bacteria in the system. And this is what Levin and Bull had done, and this is also what we had done in this concept in vitro paper. However, keep in mind that in an in vivo context, by adding phage into the lung, if a phage kills a bacteria on one lobe of the lung, it's not immediately have access to bacteria on the other lobe of the lung. So there are many ways to think about these heterogeneous mixing, and often this is just done by phenomenologically adding a saturating-like curve. So it has some sort of power law-like behavior, and this is done in spatial epidemiological models, in which, in other words, you reduce the effectiveness of mixing by saying that as viruses increase, the rate at which they come in contact with bacteria doesn't go up proportionally, but sublinearly. There could be other issues too. As you increase the phage density, if they start infecting the same microbe, you don't get more killing. So if it's one microbe, you don't get 10, just because 10 phage got in there, you don't produce 10 times as many outcomes. You don't get 10 times as many cells killed. And the way that this is modeled through these phage saturation models, that at some point they come to a harder stop, that you end up reinfecting the same cells. The point here is that I'm going to call this HM and PS, which are phenomenological models meant to represent heterogeneous mixing and phage saturation. And we're going to replace our in vitro linear model with both of these. Okay. But in doing so, I'm going to make two variants. First of all, I'm going to ask what happens in this model without resistance, going back to the question that you asked. So without resistance, here's the HM model, the PS model. We add our phage, it starts to increase, and we actually see a transient rise in the sensitive bacteria, but eventually elimination over about a 24-hour time course. So now we're in line with the time course of the in vivo experiments, and that's true for both HM and the PS versions of the model, the heterogeneous mixing and the phage saturation. And the other point here we're trying to make is that you could have used a number of other phenomenological models that have this saturating effect, and you get time courses that are far slower than this sort of miracle cure dynamics. That gets us in line, but there's a deeper issue. Again, this is a course on mathematical models of evolution. I'm not talking about viral evolution yet. But I do want to mention a very important point, which is that we've just added one virus type, and yet it worked in vivo. And given the densities of the initial inoculum of the Pseudomonas aeruginosa, there should have already been phage-resistant bacteria in the initial exposure. And yet, how did this therapy work using a single virus type? In this case, I'm going to assume there was a susceptible population and a resistant population from the outset, in which phage can infect and lyse only the susceptible kind, but not the resistant kind. But yet the immune cells can affect both susceptible and resistant cells. In that case, what you see is the following. This is HMR, but the same story happens with slightly different kinetic rates in the PSR case. Here you have a sensitive bacteria, here's the resistant bacteria. Phage are added, both are increasing, right? And at a certain point, though, the sensitive bacteria are going down, and yet resistant bacteria are present. Phage there, it doesn't target that resistant bacteria at all, so why does it go down? Because the immune system has been stimulated. So now the stimulated immune system can eliminate the resistant bacteria, and the phage have drawn down the total densities so that the negative feedback loop of having elevated bacterial density doesn't kick in. In other words, with this active immune system, the phage eliminates a component so that the sum of these is low enough that the immune system on its own can eliminate it. Okay? So what's nice about this is it provides additional robustness. You can add a phage that doesn't necessarily target everything that's in this infection, or at least resistant strains, and yet the result can still be positive. Okay. So that began to resolve that first part. How do we go from in vitro to in vivo? And we think part of this is that in vitro mixing is nothing like in vivo mixing, and we are beginning the process of trying to do new experiments to probe and disentangle which one of these is more appropriate or which combination is appropriate. The next question, though, is what is the effector cells? Yes, Paul?
Just say something about the mice, how they were inoculated with Pseudomonas and how the phage was added.
Yeah, so they were added intranasally, so it was all an intranasal addition, and 2 hours later, I'm just trying to remember the initial doses. I'm going to show some of the initial doses, but it's all done intranasally. And these are acute infections. There are different mice backgrounds, and depending on the mice background, whether or not they're neutropenic, as I'll show, or a few other cases, the doses had to be modulated to make sure that the time courses took about 24 to 48 hours for killing.
So the
yeah.
bacteria spend only 2 more hours in the mouse relative to, so I was just thinking about the structure of the environment that you are there, and really how far into the system how the bacteria go on.
Within the 2-hour case, this is where they are, even in the phage case. So they're in the lungs. They're not just in the intranasal component. So, they have penetrated into the mouse lungs even 2 hours in. And it's usually a 10-to-1 dose of phage in terms of phage particles per CFUs. And feel free to poke me more if there are things that you wanted to ask more about that. But it turns out the mixing rates is something that we're trying to get next experiments to work on, and some new models as well. Okay, yeah, right. So what are the effector cells? So one of the ways to probe where of these many immune cells are responsible for the synergy is to begin to do experiments that may bifurcate and eliminate likely candidates from unlikely candidates. And without going into all the details, one of the most important questions to ask is this likely the adaptive components or the innate components? And the time scale suggests this is probably going to be innate. What? Yes, but there's still a lot of players in the innate, which I'll get to, right? So the answer to that is yes, but you still have to show it. So this is a MIDE88 background immunity activation deficient host. In other words, mice that they have neutrophils, but they're not signaling correctly in response to the infection. And what one finds is that although the wild type case is 100% survival, when you have this new background, both the saline and phage treatments lead to failure of therapy. Okay? And you can see this both in the survival curves, but there's an interesting difference here in the radiance curves, where it looks like the phage treatment is doing well. It's almost at levels of detection, but then it rebounds, and all of a sudden it's not. So in our models, this is the HMR model and the PSR model. Let's just focus on this one. Here are the sensitive bacteria. You see the immune system is there, but it doesn't increase. So it's not signaling to draw new neutrophils to the site of infection. And what you see is that the sensitive bacteria go down, but because the immune system hasn't activated, the resistant bacteria increase and are then not able to be controlled by unactivated immune system. And we predict then that at the end, the failure here is due to the proliferation of resistant types. And when they then plate out the bacteria from the sacrificed mice, they found that all of the colonies were resistant to the phage. So we really have an uncontrolled proliferation of resistant bacteria because of the absence of this innate signaling. Okay? So this suggests that there's somehow innate effector cells. I just want to show there's something here beyond just the fact that it's innate, but that there's the failure is due to the proliferation of resistance. So we have evolution happening inside the mouse, leading to this failure of therapy. But there are many innate effector cells. And so I want to point out that it's possible to eliminate a large chunk of these innate cells, and this is done using an innate and adaptive lymphocyte deficient host. In other words, it doesn't have innate lymphoid cells, B cells, T cells. And yes, we don't think that the adaptive components are involved, but there's a major component of innate lymphoid cells. You can eliminate them, and the phage treatment is nearly 100% success. Saline treatment leads to 100% mortality. And again, it suggests that the radiance goes to near levels of detection, just as if this had been a wild-type mouse. So it didn't need certain components of the innate system, and this suggests it is likely to do with some synergy with neutrophils. So it's not innate lymphoids. Yes, there are other things like macrophage, but given what they knew about Pseudomonas, they had an idea that this would be potential synergy with neutrophils. So in this last case, this is the first slide I showed you, this anti-GR1. This is a depletion assay in which you deplete the mouse of neutrophils before the experiment. Right? So they are neutropenic, meaning they don't have these very important components of the innate immune response. And what one sees in the model is exactly the same, though a little bit more extreme as before. Now, instead of this dashed line, there are no immune components. And as you might expect, once you add a phage that affects and infects the susceptible type, the resistant type will then proliferate unchecked and reach then its carrying capacity, leading to therapeutic failure. And it's true, I haven't included the counter-resistance of the phage, and if that happens, maybe the system gets more complex. But from a therapeutic perspective, you don't want to hope that your drug evolves to a better drug somehow to make it work. Right? And so this points at a phage-neutrophil synergy as underlying this curative treatment. And this goes back and really, I think, helps to modulate how we understand those cases where phage therapy does and does not work. I want to point out that there's this question of timing, because Paul, you asked it. One of the things, and this goes a little bit to Matteo, your question too, what happens if you were to add phage in advance, almost prophylactically, and do it before the bacterial exposure? Would it be that the system just cleared out the viruses so quickly that it would be unable to be effective? So we did simulations, and they also had done an experiment where they added the phage 4 days, 96 hours before exposure. And this is in the wild-type mice with, again, 100% mortality, saline, 100% survival, even though you gave the phage 4 days before. There had been this idea that phage were eliminated very quickly from the host, and it turns out not to be the case. Yes, they're decaying. So in our model, they had separately estimated residence times, and we thought that the phage would decay, but given the doses and the fact that the phage then proliferate, they can, even though they start at lower doses initially, they are present when the infection occurs, and for the same reasons, susceptibles proliferate, but are controlled by the phage, and then the activated immune response eliminates both. And I just want to also give some immunogenic evidence that there's not statistically significant differential production of cytokines, so they don't see, it doesn't seem to be that, at least over this time period, that the immune response is actively clearing the phage, so there's not significant priming of host immunity, and you can see this in terms of LPS. So if you expose the mouse to things that are sort of chopped up cell wall that should be immunogenic, you see the cytokine response, and yet when you add phage, you don't see that response.
Right. I was wondering exactly about this inflammatory response. So when mice die, they're definitely dying, or they're being sacrificed because of the infection, not because of the inflammatory response.
Yeah, so they have active infections. This is something that people had raised, but they are succumbing from the illness, and then they're sacrificed, obviously because of guidelines on animal care. But right.
Presumably, there are concerns about the inflammatory response if you are bursting bacteria. Those pieces of bacteria will certainly.
That's right. But you would have seen, if that were the case, then the curative therapy would have led to this massive inflammatory response that would have led to, but we don't see that in those successful cases. They don't succumb to an inflammatory response. That's right. And this has been brought up also by Daniel Nelson, you might know, who used to work with Vince Fischetti. That has been another concern that somehow, and has directed some people to try to have the phage get in but not burst, somehow disable hosts, but then you don't get the advantage of the proliferation. So there's a bit of a trade back here. At least here we don't see the inflammatory response in the successful case being the issue.
With chronic lung infection, thinking about Pseudomonas,
Yeah.
if there came to inflammatory response in cystic fibrosis,
So yeah, the chronic cases are different and harder, and is also why there were strategic reasons why I thought working in an acute case was our right first move, and also places where there's easy access. So lung and burn wounds for me, I think, are the right places where I think phage therapy, given thoughtful thinking about the immune context, can be easily applied. I think with chronic, you have polymicrobial infections and a lot of other things going on. Same with gut modulation, that I've been less, anyway, it'll take me some steps before I want to go in that direction. We wanted to start in these low diversity, easy to access contexts. Okay, so those were the main parts, and I just want to bring up one last piece before I wrap up. Okay, so there's some new directions, and I'm going to go back to the work of Paul Turner, because we've begun through the course of running into each other at a couple conferences and exchanging emails and having conversations about ways in which some of what we're doing, which is focusing on synergies with immune components, and what he's doing with his synergies with antibiotics, might actually be brought together. So let me remind you again, at the very beginning I mentioned it, so now you see a blown up view. There are antibiotic-resistant Pseudomonas aeruginosa, which are phage sensitive because they pump out the antibiotics but let in the phage. There are also antibiotic-sensitive Pseudomonas in which they don't have these efflux pumps, which means the phage can't get in, but when you put the antibiotics in, they can't pump them out. Right? So this evolutionary trap is what they use to cure this individual and are continuing to push ahead. When I heard these talks, I was a little bit worried that was there some level which, even though this seems quite appealing, that the immune response may still play a role even in these cases, and is there a way for us to think about that in addition to the dual therapy, because if it fails because of the immune context, there are other ways for us to deal with it. So let me go back to the initial model, which you've seen now in depth, these phage-sensitive inoculum. I think, Marcus, is your question. If the inoculum is dominated by the right type, your target host, then yes, when you do these phage addition in the models, it eliminates the sensitive bacteria, resistant then decline because of the immune. But if the resistant type you've mistargeted is at higher abundance than is the sensitive type, then the phage are going to decay, the resistant bacteria increases, immune system turns on, the phage can't get at this one, and this one inhibits the effect of the immunity, and so you have failure. So it really depends, and there's a critical density at which this switch happens. So you have to really know that you're targeting the right type, and there's some fragility here. Okay. And the reason why we think this is relevant is because if we want to go then in the context of what Paul Turner's doing, they're considering these two types, the antibiotic sensitive and resistant, by adding not just phage, but phage and antibiotics. So this is a schematic in which we have two bacterial strains. They're not just sensitive to phage or resistant to phage. They have two properties, antibiotic sensitive and phage resistant. So BA is sensitive to antibiotics, and BP is sensitive to phage. So I'll use the second letter to denote what it's sensitive to, right? BA and BP, sensitive to antibiotics or sensitive to phage. And you can see the phage kills that which is sensitive to phage, and the antibiotic kills that which is sensitive to antibiotics, and the immune cells work on both. So we've just taken our model and added this little component, okay? And so what we find is that if there's a phage-sensitive inoculum and we add phage, it's going to work. But if the system is dominated by these antibiotic-sensitive inoculum, which are resistant to phage, then phage therapy fails. And yet if we have combination therapy, then we find that in both cases we have success. Okay? So this begins to suggest, in a model version, we're really just repeating what it is that Paul had done and showing the evidence for why this seems, and I think it should be evident why it seems to work. But this is the twist, which is that here we have this combination therapy, phage-sensitive or antibiotic-sensitive, with a immunocompetent host. So we've taken our model, added the antibiotics, and shown that irrespective of what the inoculum is, whether it's resistant to phage or resistant to antibiotics, that this therapeutic addition will work as long as you're adding both phage and antibiotics in an immune competent background. In contrast, if you do the combination therapy, the thing that Paul suggests one should do, which is phage and antibiotics, but in an immunocompromised context, then it doesn't work because yes, you can hold down the antibiotic sensitive type with your use of antibiotics, but the phage and bacteria are then going to coexist, and you're not actually going to eliminate the bacteria. You get a switch in composition, but you don't get elimination. And so our thought here is that this suggests that phage and antibiotics may still, even though they're going to be more robust, may still require some level of immunocompetency for them to be effective. Okay. And just to make this point out is that this is a generalization. On the x-axis, we try different levels of inoculum. So antibiotic sensitive from no antibiotic sensitive to all, treating it with antibiotics only or antibiotics plus immune, and then looking at the antibiotic levels. This is the minimum inhibitory concentration. So this is lethal levels. These are sub-inhibitory levels. And we find when it's only antibiotics, what we do is a selection experiment, and we get a lot of phage-sensitive bacteria. When the immune system is there, it turns out there are regimes in which it can actually eliminate the bacteria, but it's still largely a selection experiment, and you end up with the phage-sensitive bacteria. When you do antibiotics plus phage, irrespective of the initial conditions, you get phage-sensitive bacteria when you use a lot of antibiotics, or antibiotic-sensitive bacteria when you use less. And our point here is that when all three come together, you get elimination in this regime when you use lethal doses of antibiotics, but you also get curative efficacy in sub-inhibitory levels, which is very important because people would like to use sub-inhibitory levels to minimize the chance of evolution of antibiotic resistance. So our hope here is that this could guide not only the levels of antibiotics used, but also provide some context for deciding who is a good candidate or what does one need to think about on the immune side in order to use phage and antibiotics together for curative treatment. And I should just point out that this is work in prep. This doesn't involve in vivo work yet. Right now this is an in vitro model taking these in vivo parameters and evaluating what happens in different immune contexts. And just to have one last point, this is an opinion article. It's one of those rare opinion articles in which my opinion is couched in mathematical language, so it's a modeling paper couched as an opinion, in which we try to generalize this term too. And I just want to point out, in case you're interested, it's not about phage, but we have a pathogen, an immune response, and we swap out the phage for a commensal and ask similar questions. So if you're interested in microbe-microbe interactions, our effort here is to try to say that more generally, we saw phage immune synergies, phage antibiotic immune synergies. There could be a generalized feature of the use of therapeutics, which is not necessarily on their own to eliminate the infection, but to enable the immune system when activated to eliminate the infection. So if you want to read more, we get some of the similar results where commensals alone don't do it. You see the sort of scary pathogen increasing. The immunity alone doesn't do it. But similar to the immunophage case, we have the replacement of, or the displacement of the pathogen by the commensal as long as there's immunocompetency. Okay, and that's the last slide. Let me just get to the conclusions now. I introduced this tripartite model of phage, bacteria, and the immune system. In other words, taking phage bacteria dynamics, the ecology and evolution of that system, and putting them into a different environment, an immune host in which there was an immune context. And the analysis showed that curative success, and this is even without modeling, it's clear the in vivo data shows that it depends on both, not just the phage, but the phage and the immune response. And that both are critical for curative success. Further probe of the experimental system showed that you can that there's a phage-neutrophil alliance, or there's a synergy necessary for therapeutic success. I also want to point out this begins to resolve this resistance problem. One doesn't need to necessarily have phage to target every possible strain. Insofar as there's a robust immune response, it's possible that subpopulations can arise and still be eliminated, even though we haven't directly targeted those potential subpopulations. And my hope is that we'll begin to explore these generalized notions of synergy, exploring interactions between phage, antibiotics, and commensals with the immune system to come. And so that's what I talked about today. And just if you want to read more, this is the in vitro model. This is the in vivo model. This is a modeling paper as an opinion. And we're still working out some of the last details of this phage antibiotic immune synergy. And this is the fourth of the lectures. I tried to go from ecology to evolution, from lysis to latency, really contrasting these ideas of obligate killing to those in which latency might be relevant. And this last bit showing you some opportunities to take what we know about phage ecology, microbial ecology, and put it in not just to an environmental context, but into a human health therapeutic context. And most of all, thank you for listening. Really appreciate it. I'm happy to take some last questions. I know it's late, and probably time for lunch. Thank you.
Questions?
I have a curiosity, actually. In the beginning of your lecture, you mentioned that some people thought about put a lot of more than one kind of phages.
Phage, yeah. Yeah.
Thank you. Can these different kinds of phages present interactions between them? Related with, like, do the phages interact between them?
Yeah, so I'd say that the use of the cocktails is almost predominantly a selection of phage that can target different bacteria rather than hoping that two phage interact with one cell and together lead to elimination of the cell, right? So if that were the case, there's other issues that might come into play. It's not to say that can't happen, but right now, the choice of phage is often because, in the cocktail sense, you have a target microbe, and you expose that microbe to phage, you'll have one-step resistant mutants, and maybe you could then isolate phage that infect those mutants. That would be a procedure or recipe to generate a cocktail in which your attempt is to find a variety of phage that target different variants that you think are likely to arise, rather than to get two phage, kill one cell synergistically.
Thank you.
Okay, thank you for the nice lecture and course. And I have two questions. The first is that you have a dynamical system, and you have lots of parameters. You said, oh, we took parameters from vivo, but to me this is really difficult. I had some problems with that one time, and well, and we have lots of data, but how do you fit that? For me, it's amazing.
Yeah.
And then,
so,
sorry.
This is why, first of all, I'll say that this is why the process from which we spoke to them in the summer of 2016 to when we got these models ready, the bulk of the time was actually going through carefully and trying. These are not all fit. There's so many things. If you just fit these, you're eventually going to get something that works. But we're using the doses that they had. We then fit some of their neutrophil recruitment data. They did other experiments so we could get residence time of phage. This is a laborious process, but it turns out, I feel that particularly the concept was there. And at the end of the day, I think the biggest thing we learned was that, let's say from the right physics of the system, is that the mixing is almost certainly not well mixed. Now, what is the exactly right function or parameter? It turns out that probably different variations, as I showed you. We tried to intentionally because we don't know which one is right. We have a proposal to try to figure that out. If it gets funded, then we have to do new experiments. But a lot of the time it took was Joey talking to Dwayne, working through, getting on the same page about language, and trying to even establish what these correct parameters were so that the fewer sets of free parameters were then far more restricted to what we think was the interesting unknowns rather than the stuff that is the background that we should really just have the in vivo data for. But it took a long time. Sorry, I interrupted your half question, but...
No, the other, one picture of the bacteria being attacked by the immune system, it resembles a biofilm. And as if the bacteria had a spatial structure.
Yeah.
And if this is true, I think that maybe a mean field would be problematic. Thank you very much.
So I agree. I don't know if that was a question, but I'll turn it into a question. Is it appropriate? I think the answer is no, and I think I agree with you, which is that which is why we chose this acute system, which we think density dependence and the timescales are not going to establish the kind of complex biofilms in a CF lung, in which then spatial dynamics are going to be much more important. There are some reasons why still I think we need to explore some of the spatial dynamics even in this model, and that we're trying to go from a microscopic model to see, do certain kinds of microscopic interactions lead to these phenomenological models when we then coarse grain? We haven't done that work yet, but that's what we're hoping to do. Yeah.
That was going to be my question,
Okay.
yeah. So all this, when you write things in terms of differential equations, there is this hypothesis behind that everything interacts with everything else.
Right.
So you think that is an issue that could be important to...
Yeah.
I think when you look at the mice, you see these spots, and things are not really spread out uniformly.
Yeah, so there are two levels in which there's heterogeneity. First of all, there's just, even in a homogeneous spatial environment, the fact that there's discreteness and spatial localization could mean a different kind of dynamics when coarse grained. And so we have in other work looked at phage-bacteria models in space, and what you find is because a bacteria reproduces, if a phage infects and lyses a bacteria, they tend to be clustered, which means you get elevated levels of multiple infections in a way that's higher than you expect from the mean field. And that can lead to, and that would lead to saturation. But the mouse, and so we have some in vitro work that we are working on. So we have a funded project to do phage-bacteria interactions in homogeneous environments, not like the lung. And then we are hopefully going to try to push up this lung work, but that gets, you have to section things, it gets much more complicated. Yeah.
Thank you, Joshua, for the nice course. I was wondering, relating the previous talk to this one, can, in this kind of context, the lysogenic response is relevant in AHA?
Yeah, so I should point out that there's a choice of what kind of virus to use when you're doing phage therapy. And almost exclusively, the field has gone in the direction of obligately lytic or virulent viruses for the reason that they would like to get the benefits of the killing and not have, by accident, the phage integrate, not eliminate the bacteria. Now, there's been some reasons to think that that could change, but at the moment, and everything I've shown you here is obligalytic because it's an obligalytic phage. And to the extent to which one has a temperate phage, there are ways to modify them to eliminate its potential to become a lysogen. That's not to say there couldn't be some gene transfer event, but for the moment, everything I said here is more related to, in fact, I thought about switching the order because I introduced a latency before. Now I'm back at lysis again. But I also thought that actually the order here was the right order, and I wanted this to be the application side. But it connects to the earlier lectures. We're still in the lytic mode, not the lysogenic mode.
So in your model, you have like a mutation rate in which a bacteria becomes resistant. Did you explore how sensitive it was to model to the mutation rate changing?
Yeah, so those are a component of the model. They are small parameters because these aren't genotypic mutation rates. They're not the chance of having a particular mutation. They're actually a phenotypic or effective phenotypic mutation rate. And so those are relatively well established from Paul Turner's group now. But anything that is small, what's driving the abundance changes is not the fact that new mutants are being generated, but rather the new mutants are proliferating, right? Now, the negative of the way we've approached this, in order to resolve the fact that if you paid attention to the first lecture, you know the Lurio-Delbruck experiment, they're already there, and the abundance may be variable at the very outset. So we assumed always the same concentration of resistance. But the phenomena, because it was always going to be so low, as long as it's low at the beginning, then what I told you holds. As I showed you later, if we just totally mistargeted, but that's not because of mutation anymore. That's because it's either already grown or it's mistargeted. Okay. Yeah.
The slide with the data from the experiments, I mean, there are many things there that are interesting, but what is known about the relationship between the initial inoculum density and subsequent effects? And I guess I'm asking that because I'm wondering, given that massive load of bacteria and the even massive dose of phage, how much of the clearing is dependent on secondary infections, so amplification of the phage, or is it basically a one a one-hit shot from the phage, relying on no subsequent amplification of the phage through the bacterial population?
I think it's a great question. I think that the 96-hour pre-experiment helps a little bit to suggest it's an amplification component.
Yeah, that must
because they did do the clearance, and I think if I went back there, we can even look at the decrease in that density, but it goes down multiple logs in this, where is it? Where was it? This is the case. We're, in this case, going down to a case where we think it's not a single shot hit anymore, if our clearance estimates are right, and they seem to be reasonable. So yes, it started off above, but then went down to at least a level that it was about the same or even lower. I agree with you that you saw the early work, if the phage density were initially too low, then we didn't have curative treatment, which suggests you have to get into some initially to be able to kinetically keep up with the growth of the bacteria. And so my instinct here is that even in the case where it's lower, it's amplifying, but just because of the growth of the bacteria, it's taking too long and the mice succumb. So my feeling is, though, I think that you could do this directly by modulating the phage so that it gets in and kills, but you could eliminate the lysine or the holin, or you could modify it so it doesn't actually amplify, it just kills the target. And then see if you get the same effect. So I don't have direct evidence along those lines. This is my one indirect evidence that that my instinct here is that there is the amplification that plays a role.
Yeah. Well, it's quite interesting that the phage can last so long. I mean, its decay rate is relatively slow, so there is the potential, of course, for it to amplify, given that it's not immediately, eliminated. If it's
so that is it's a feature here, and this has been a big debate in the phage therapy field. Also, when you're adding things to blood versus lungs, it's very different. So the clearance rate, the fact that it's in the lungs, I think that's also, these clearance rates are with respect to the intranasal application. They're not saying that if you applied the phage somewhere else, it wouldn't be eliminated more rapidly. I think that's an advantage here.
Any other questions? Okay, so I think, there's one more.
Last question about, so you said this combination of immune system and phage therapy can solve the problem of antibiotic resistance. So, the
resistance, let me clarify.
Yeah.
The resistance problem I was talking about there is the resistance of the bacteria to the phage. So.
Yeah, okay. So now.
And also, we should be very careful. These are mice.
But this is going to evolve too, probably, no? And so what do you think, would this be, will there be another race on phage resistance?
So that race has been going on for a long time, right? I think so. What did someone say in one of the seminars? I mean, this is a billions-of-year-old race. And so we have that's the other advantage here. We have quite a lot of. No, I'm talking about phage-bacteria race. Right. So the resistance, and really the point I'm trying to make here about thinking about the immune context is that a lot of the development of therapeutics focuses on envisioning that the way that therapy works, the way the antibiotic works, the way a commensal works, is that thing that you add is the responsible thing. And our point here is that you've added to a system, and your objective is to move the system from an interior place to a boundary. And our view is that even though we designed a therapy only to target a component, we're able to move it all to the boundary. And that's what I mean by solving the resistance problem of the bacteria to the phage. We didn't have to design. I think that should give some confidence, but it also changes the perspective. So for example, if I think what I need is phage that kill effectively bacteria in vitro, that'll be one way of doing my therapeutic development. If I think what I need is phage to eliminate bacteria so that the immune system can access the bacteria, that is a different lens for development. Maybe I need phage that can penetrate biofilms even if they don't have as great killing activity, because that thing will help the dynamical system move it. So I think that's the other thing that I'm trying to change here. the perspective of what are the phage doing? Do we want the best killers or things that will enable the immune system to take over? I think it's the latter. I could be wrong. It's probably a mix of both. And killing is part of that aiding as well. Yep. Oh, one question.
I know that you mentioned that the response from the phage, so like which would be like the co-evolutionary side, is not the main goal of those therapies, but how common is that, or like do people know how common is that, and how would you incorporate that in a model?
So I think you're right, and that was the point I was conveying, that it is not the central interest of those who are trying to do phage therapy. And in fact, when we were trying to talk to the group that we collaborated with, we wanted to do the co-evolutionary model, and they were not so interested in the co-evolutionary model because they didn't see that as a route to getting a therapeutic. Clearly, co-evolution happens from whether it's in these in vitro cases or in vivo context. I don't think we quite know the rate at which there might be already phage in these succumbed animals that could be able to infect the resistant bacteria. There's a density problem, right? Whereas if the sensitive bacteria are eliminated, you have largely resistant bacteria, the viruses have to get into some cells to reproduce, to generate variation. And so I just, at the moment, don't know how much of that is happening at this time scale, if that's the right reservoir, or if the better thing to do is to find the resistant bacteria and separately hit them with as large a dose as possible of phage to identify the host range mutants. And I think that latter case, you probably also don't need to use the mouse necessarily. You have a higher chance of success. So I'm interested in the co-evolution question, but from the therapeutic side, I'm thinking more about how do phage, a limited subset, still work, even if the potential range of bacterial hosts that are inside the host is larger. Yeah.
This virus can invade normal, regular cells in lung.
Bacteria. So this virus can only infect Pseudomonas aeruginosa.
Only.
It's not infecting lung cells.
So the immune response isn't because the virus invade also lung cells.
Right. So this is this next slide. If you only add phage and you don't add the bacteria, you don't see an immune response. And also, remember, viruses infect many things. Yes, viruses could be circulating in the lung, but they're not actively replicating inside human cells. They are only infecting, replicating within bacterial cells, and in this case, only within the Pseudomonas aeruginosa. Yeah. Okay. Can I say thank you? And we can close up. Any more questions, or are we good? Oh, no, we have one more. Daniel. I think it's appropriate Daniel should have the last question.
So, I mean, it's maybe, yeah. So there's a huge social aspect of this also. So my question would be, like, what's your perspective and what sort of, yeah, like what's your estimate of when we have phage therapy on society? I mean, there are lots of changes that have to happen first, of course, but what's your perspective on that?
That is a very relevant point, that there are certainly reasons why, given the historical record of the lack of repeatability of phage therapy, that many people are concerned. And also there's other forms of biases in which it was used in one place. So former Soviet Union breakaway republics like Georgia and so on, and wasn't used in the West, so therefore maybe it doesn't work. There are other issues, irrespective of that kind of baggage, like do you want the FDA, the Federal Drug Administration, to approve the use of a therapeutic a therapeutic virus, which from the perspective of the public, if you use the word phage is going to sound like antibiotic or some other molecule. But eventually people will say, oh, do we want to add something that replicates inside us, right? And then there can be concerns. What happens if that phage mobilizes a gene and then something else happens? Now, of course, phage are inside us doing that already. So I think that there's a push and pull. We need to do a better job showing and demonstrating what are the conditions when phage therapy works. Because also, when there's a drug trial, the kind that was done in Europe that had a lot of fanfare, and it doesn't work because you don't have the inclusion criteria correctly, that impacts the social perception of the value of doing phage therapy. Also, if you try to, there's always these examples of the phage therapy that works. What happens to the ones that don't work? I mean, certainly, it's not going to work all the time. Those efforts could be because it was just a difficult case, a lot of comorbidities, or because we didn't understand the curative dynamics of phage therapy appropriately, and if we had, we would have seen, the immune context isn't right, it's probably not going to work there. Why are we using that in that context? So I think that's the job that we have to do on the science side. At the same time, unfortunately, the need is increasing, and so that's also going to make people maybe more likely to consider it. So I think we have those two things happening at once. It's an unfortunate timely coincidence, right? We'd rather not have these multi-drug resistant problems, but now that we have them, I think we need to do a better job of understanding what are the conditions when phage therapy works and try to bring those ultimately to a clinic setting, but carefully, right? So, thanks.
Okay, so thank you, Josh. Thank you very much for the lectures. So, the school is officially closed. Thank you all for coming. I hope you have enjoyed the lectures. A special thanks to our lecturers that took the time to come here and to prepare this very nice series of lectures. So, thank you all.
And maybe we should also thank the organizers, I believe. Marcus, Matteo, who are the major organizers? And Roberto, maybe we should thank them as well for organizing this.